Very good morning, students. Welcome to biology classes. Today we are going to see 11th science biology, and we are going to see the chapter number 8, that is cell, the unit of life. In this chapter, we are going to see the details of cell. This chapter you have already been completed, right? In the previous, you have learned about this chapter in previous standards, right? But here in 11th science, when you're going through this chapter, you are going to see the details of different types of the cell organelles as well as the cells. Okay, now, so let's start with today's lecture. Okay, now all organisms are made of cells or aggregates of cells, right? Now see, you know that every single organism is made up of cells, right? And all the organisms, they are made up of cells or the aggregate of cells. That means there are so many cells which are present, like they are attached or combined with each other to form the aggregates, right? The cells, they vary in their shape, size and functions, right? So the cells are of different types. They vary in shape as well as also they vary in size. That means the size of all the cells is not same, right? Every size or every cell have different shape and size. Also all the cells are performing different functions, right? Next is, based on the presence or absence of a membrane bound nucleus and other organelles, cell can be named as prokaryotic or eukaryotic. Now the first main basic uh, division which is observed, right, is the prokaryotic and eukaryotic. But this prokaryotic and eukaryotic, it is based or they are separated on the basis of the presence or the absence, right? Whether a membrane bound nucleus is present, whether membrane bound organelles are present or not. So the first main difference between the prokaryote and eukaryote is what? That the membrane bound organelles and nucleus, right, either they are present or not, right? So we are going to see in detail about the prokaryotic and eukaryotic in the further lecture, right? Now, the cell is said to be the fundamental structural and functional unit of all the living organisms, right? This definition you have been studying since 7th or 8th standard, right? So the definition of cell is what? It is the structural and functional unit. That means the cell imparts the structure, right? As well as it performs its specific function, right? Say, let's take an example. The heart, the cells of the liver, right? They are going to secrete what? Only and only bile juice. It is not going to secrete any other secretion, right? So that is the function of the hip, like hepatocytes, that is the liver cells. Same way, it also, the cells, they together form or they together join with each other to form a structure. They are going to impart a structure, right? For this reason, cells are said to be the structural and the functional unit of all the living organisms. Now, Anton von Leeuwenhoek was the first who observed and described a liver cell, right? So the Anton von Leeuwenhoek, he was the first one to describe the liver cell, right? He was the first to observe the liver cell. After the Anton von Leeuwenhoek, Robert Brown was the one who discovered the nucleus, right? Next is, now we are going to see about the cell theory, right? So what the cell theory says. So in the cell theory, Schleiden and Swan, right? They were the one who gave the theory of the cell. That means what are cells and how the cells work, right? What exactly the cells are made up of, right? And after Schleiden and Swan, later on Virchow, right? Virchow was the one who worked on the theory which was being given by Schleiden and Swan. So initially the first one were the Schleiden and Swan who gave the cell theory, and uh, based upon the investigations and the information which was gathered by the Schleiden and Swan, later Virchow worked on this theory, right? So all the living organisms are made up of cells and their products. So what they found is, upon gathering the information and working on the project of the cell theory, what Schleiden and Swan and the Washoe, what they said, that all the living organisms, right, they are made up of cells and their products. So there are several products also which are being produced by the cells, right? So all the living organisms, they do possess cells and their products. Then all the cells, they arise from the pre-existing cells. They also say that the cells are not developed, right? They are being formed. They are arising from the pre-existing cells. That means already initially a cell was present, and from those cells only, upon carrying out the division, they are forming new cells, right? For this reason, they said that all the cells are arising from the pre-existing cells. Now, the first type of cell which we are going to see is the prokaryotic cell. Now, you can see here, you can see this figure also, right? There is presence of a flagellum, this red colored dots which are observed in the whole inner area, that are those are ribosomes, right? In the middle portion, the light blue color you can see, right? This blue color, that is the nucleoid, that is the genetic material. The green colored lining where I'm showing the cursor, this is the plasma membrane. After that, a yellow colored layer is there, which is the cell wall, and an outer orange colored layer, that is the capsule, right? Normally, you know that prokaryotes, under the prokaryotes, bacteria comes, right? So this is also a kind of bacteria, right? There is also presence of, here I am showing the cursor, the blue colored small thin thread like structures. These are plasmid, right? That means those are the extra chromosomal DNA, right, which have the ability to replicate themselves. Now you can see on the surface there is presence of green colored villi, right? Right, this they what they do is they uses this for the locomotion purpose. This flagellum is also useful for the locomotion purpose. The inner fluid which is observed where I'm showing the cursor, right, this all is said to be the cytoplasm. Cyto means cell, plasm means the fluid. So the fluid of the cell is known as cytoplasm. Now bacteria, blue-green algae, mycoplasma, then PPLP, that is pleuropneumonia-like organisms, right? This all are coming under the prokaryotic cell. That means this all are prokaryotic cell. Amongst this, there is an main thing which is I would like to tell is the mycoplasma, right? You have seen in the earlier, the first unit, right, that mycoplasma are the smallest living cells, right? So this can be asked in the MCQ. Next is glycocalyx, cell wall, plasma membrane. They are present, right? So here the cell wall, see all the prokaryotes, they do possess the cell wall as well as the plasma membrane. Along with that, they are also containing glycocalyx, but it is not present in all of the bacteria, or we can say all of the organisms, right? In few organisms only it is present. Now, based upon the staining property, they are being differentiated into gram positive and gram negative bacteria. So when we are talking about the staining property, that means the bacteria are of different shapes, right? And to understand them, or we can say if you want to check that whether there is any presence of bacteria in the medium or not, then you need to stain them. Stain them means you are using several dyes to color this bacteria, right? So that is the staining property, right? So based upon the staining, you are getting two different types of bacteria. The first is the gram positive bacteria, and the second one is the gram negative bacteria. There is also presence of the mesosome, right? Also chromatophores are present, right? That means the chromatophores are the extension of the plasma membrane. Mesosome is initially present in the cell, but it is found in the prokaryotic cell. Next, chromatophores are the extension of the plasma membrane. That means several extensions will be observed which are occurring from the plasma membrane, and they are known as chromatophores. Now, this type of bacteria are, some bacteria are motile, that means they can move from one place to another, that is simply locomotion, while some bacteria are also there that they are non-motile, that means they are not able to move, right? Now The flagellum, they do possess three parts, right? The flagellum is made up of three parts: the filament, hook, and the basal body. Basal body is the first thing which is attached to the cell, right? That is a bacterial cell. Then there will be presence of the filament, that is the thread-like structure, and at the last there will be presence of a hook-like structure, which which like takes the help of any of the substratum and they move, right? So that is the three parts of the flagellum. There is also presence of pili, right? Then fimbriae, right? These two of them, they are the surface structures, do not play a role in motility but helps in attachment. As I said, this pili and fimbriae, they are not responsible or they are not useful in the locomoting, right? That is passing from one place to another. But simply, this pili and fimbriae, they are responsible for attaching to any of the surface for getting the support, right? So for the attachment purpose, pili and fimbriae are used. There is also presence of ribosomes and some inclusion bodies, right? The ribosomes, the function of ribosomes is to synthesis protein synthesis, right? That means they do possess the, or they are going for the production of the protein. So the ribosomes here, several subunits and all these things have been written. That is, their size is 15 to 20 nanometer. That means they are that much small. They are having two subunits, a large one and a small one, right? The large subunit is the 50S subunit, and the smaller one is said to be the 30S subunit. So the ribosomes, they possess two subunits, 50S and 30S. When they collectively join with each other, it forms a 70S ribosome, and this 70S ribosome is useful in the protein synthesis, right? They are also going for the synthesis of polysomes, right? Or polyribosomes, right? Which is present on the mRNA. So first of all, the mRNA is there, and from this mRNA, they are getting several information, and based upon that, the protein synthesis has been carried out. The inclusion bodies are what the reserve materials, right? The inclusion bodies means wherever the food is been reserved, that is the phosphate granules, cyanophyllene, or glycogen granules. So these are different: phosphate granules, cycophyllene, or glycogen granules. These three are said to be the reserve materials which are present in the prokaryotic cell in the form of inclusion bodies, right? There is also presence of gas vacuoles, in which also, you know, that vacuoles is, they are playing the role in the storing of the food materials or the water or anything which is needed, right? So those gas vacuoles are going to again store this type of things. Next, there is presence, you can see here, right? There is presence of, okay, so next is the eukaryotic cell. This eukaryotic cell is consisting like of protist, fungi, plant cell, and animal cell, right? So the protist, fungi, this we have seen in the chapter number 2. That is when we saw about the Whittaker's 5 kingdom classification. In that there were several eukaryotic cells also, right? That is, they were coming under the protist, fungi, plant cell, as well as the animal cell, right? This, the main difference in this which is observed is the cell membrane, right? In the prokaryotic cell, there was presence of the plasma membrane. Here main is the cell membrane is present, and based upon the cell membrane, the fluid mosaic model has been given, and this fluid mosaic model was given by Singer and Nicholson, right? And this was given in 1972, which is based upon the bilipid layer of phospholipids with two types of membrane proteins called peripheral protein and integral protein, with cholesterol, glycolipids, and glycoproteins. Now see, this cell membrane, what they do is they are possessing phospholipids, right? Bilipid layer, bilipid, split the word bilipid, bi and lipid, right? Lipid means nothing but the fats, right? Bi means two. So there will be presence of two layers of phospholipids, right? That means on the upper side and on the lower side. So there will be presence of two phospholipid layers, and these two of the phospholipid layers, they are possessing two types of proteins. First is the peripheral protein, and second is the integral proteins, right? These two of the proteins have a proper function in the cell membrane. They are responsible for the exchanging from in see whatever is being needed to be exported out from inside the cell, then this proteins get opened and they send the particular things from inner side on the from inner side to outer side, right? See, you can see here, uh, what you can see is the glycoproteins, glycolipids and cholesterol. This three of them, they are useful, or we can say they are helpful in maintaining the structure of the cell membrane, right? So the peripheral proteins are present on the periphery and the integral proteins are present in between the two phospholipid layers, right? Integral means the in between and peripheral means on the periphery. So these two of the proteins, they are responsible or they are helpful in passing of the ions or other proteins from, if a protein or if a calcium ion is needed inside the cell and if it is present on outside of the cell, then with the help of this peripheral protein and integral proteins, the outer calcium ions are passed to the inner side of the cell. So this is about the eukaryotic cell. You can see here, this is the diagram which is showing two different types of cells. The first is the plant cell and the second is the animal cell. The main difference between them is the presence or absence of the cell wall. In the plant cell, there will be presence of what? The cell wall. While we are talking about the animal cell does not possess cell wall, right? They only possess the plasma membrane. Here you can see the several, I have the labelings are already observed to you, right? You can visualize it. So these are all the organelles which are present in the plant cell, and here these are all the organelles which are present in the animal cell. This all things we are going to see in detail in the upcoming slides, right? Now, the first is the cell wall, right? So before going to the main topics, right, first we need to understand which are the main cell organelles of the plant as well as the animal cell. So the main and the common ones are the first, let's start from the nucleus, right? So the nucleus is present in both of the cells, right? And this nucleus, it is containing nucleolus, right? This nucleus is going to contain what? The genetic material, that is the DNA or RNA or whatever it is, right? So whatever genetic material, mostly the DNA is there, that is present inside the nucleus, right? There will be presence of nucleolus, just like the cell is containing a nucleus. Same way, the nucleus is also containing a nucleolus in which the uh, genetic material is being embedded, right? Next is there is presence of Golgi apparatus also. This Golgi apparatus, it is going to form several vesicles and they are responsible for passing the produced things from inside to the outside of the cell. So for the passing mechanism, Golgi apparatus is responsible. Next, there will be presence of a layer, or we can say a membrane, which encloses the nucleus inside it, right? So that is what the nuclear envelope. So that is observed in both of the cells. Here also the nuclear envelope is observed. Here also the nuclear apparatus is, or nuclear envelope is present, right? Next, there will be presence of plasma membrane in the plant cell as well as it is also present in the animal cell, right? So these two of them, they are both are containing what? The plasma membrane. Here, when we are talking about the plant cell, there will, you can see this yellow larger organelle which is known as vacuole. This vacuole is not present in the animal cell. So the first main difference between the plant and the animal cell is the plant cell does contain the vacuoles, but the animal cell does not contain the vacuoles, right? Next is there is presence here, middle lamella is there, right? So this middle lamella is what? There will be presence of a fluid like structure or a small gap or space like structure between the plasma membrane and the cell wall, right? And that is what the middle lamella. But here when we are talking about the animal cell directly have the plasma membrane. They does not possess a cell wall. For this reason, the middle lamella is also not present in the animal cell. Next, the cell wall. It is only present in the plant cell. It is absent in the animal cell. Then there is presence of the ribosomes which are present in both of the cells, that is as well as in plant as well as in the animal. The function of the ribosomes is to do the protein synthesis. Next, in the plant cell, there is presence of the chloroplast, and this chloroplast is also one another difference which is seen in these both cells. This chloroplast is present only in the plant cell because there is presence of a specialized pigment which is known as chlorophyll, and this chlorophyll is going for the photosynthesis process. That is the process of producing the food with the help of the sunlight. But when we are talking about the animal cell, we are not the ones who are preparing their own food, right? For this reason, this chloroplast is not absent in the animal cell. Then cytoplasm, that is common in both of the cells, that is the fluid of the cell is present in both of the cells, that is plant as well as in the animal cell. Then peroxisome is present in both of the cells, right? That is, there is presence of small dot like structures where you can see here also peroxisome is present in both of the cells. Then there is presence of the microtubule, right, in the plant cell, which is not observed in the animal cell, right? So this microtubule is also one of the difference which is observed in the plant and the animal cell. It is present in the plant cell, but it is absent in the animal cell. Then there is presence of the plasmodesmata. That means as this is containing plasma membrane as well as the cell wall, there is presence of several, you can see here this line like structures, right? That means directly from the cytoplasm there will be inclusion which will be opening to the outside of the cell wall, and that is nothing but the plasmodesmata. So whatever things are being needed to send out of the cell, that is being sent out with the use of the plasmodesmata. This is absent in the animal cell, right? Now there is presence of the two types of endoplasmic reticulum. First is the smooth type and the second is the rough type, right? So rough endoplasmic reticulum means it contains ribosomes on it, and for this reason it is known as rough endoplasmic reticulum. Smooth endoplasmic reticulum means there will be absence of the ribosomes on it. This is seen in both of the cells, that is plant as well as animal cells. See, here you can see the smooth endoplasmic reticulum, which is a Y-shaped structure, and the second is the rough endoplasmic reticulum, which is containing dot-like structures on it, and that is the ribosomes. There is also presence of the lysosome. This is also observed in both of the cells, and lysosome means the lysis is when the lysis takes place, this lysosome enzymes are being used, right? So this is all about the different types of the cell organelles which are observed in the plant and the animal cell. There are some differences which are being observed, that some are present in plant cell but not present in the animal cell. So these are the main differences between the plant and the animal cell. I hope you are very clear with today's topic, and I hope to see you again. Thank you.